Okay, so basically what I'll do today is question and answer. Let me say, I can basically tell you the exam format though first, to be clear there. So, I for sure the exam will have 40 multiple choice questions. 40 multiple choice questions, what that's supposed to say. On this, I would say that around 10, or let's say at least like 8 to 12, about each of these topics. Questions about, this would be like math, this would include unit conversion, scientific notation, graphing stuff, like not you making a graph, but you reading a graph. Alright, so we have about a quarter of the test about this stuff. This might also be, I guess, serial dilutions. Serial dilutions. We'll have, the next topic, we'll have like 8 to 10 questions, 8 to 12 questions about it, right around 10 though, would be images. And so I guess the images I would consider either the images shown on this page. You're responsible. You should be able to talk about everything except plate Z on here on this side. So the images and this would be all but plate Z, and this might also include like a graph. I guess I was kind of counting the graph interpretation as math and images. I just realized. Okay, then I expect to have 8 to 12 questions, around 10 probably. They're straight out of the book. So they'll say like, from your lab manual. I can't spell manual, but from your lab manual. And so those would be from labs, like applicable parts from labs we've done. They're all going to have a real right or wrong answer. That it should be in the case that like every one of you has the same answer, not like what did you have or what did you have, something like this. Okay. Then the other 8 to 12 questions, I guess, that I'm left with would probably be stuff like definition things. It could also include safety things, like lab procedures or safety things. Might be like, what's the definition of resolution or ubiquity or colony morphology or things like this. All right. So I'm going to say etc. So I want to make really clear to everyone, so there's not like some people that know more than others, about the lab manual questions. So if you have questions about the multiple choiceable lab manual questions, you can review the stuff on Google, you can look it up in your book, you can talk to classmates, etc. But someone said, what if I thought I got it right, but I was actually wrong? So I was like unaware that I was going to get it wrong. We didn't consider that, so when we grade your lab manuals, we're usually just looking for completion. If you want to see the true answers according to the lab book authors, in the front of every classroom, the teacher's desk is a teacher's manual, and you can open this up, and it has like the worked out answers to the questions. You're welcome to look at this stuff and check your own, as long as you tried to answer it first. As long as you thought about the question, you can grade your own. This will only be worth it for the multiple choiceable questions, right? This might be worth it for that. Okay. What else do you need to know? So I said 40 multiple choice questions. It's all going to be answered on a Scantron form. So pencil is required. This will take place in lab, the last part of lab. You are allowed a calculator. You're not allowed like a cell phone that has a calculator on it. A dedicated calculator is okay. You don't need a calculator. I won't use one today doing the math, so it's allowed. Okay. With that said, any questions? One other common question: what's good review material? Best review material probably would be. So, what's the easiest way for me to write an exam question? It's to change an old exam question. So, which of the following is gram positive? Now you say which of the following is gram negative. Just change it like that. So good review material would include quiz 1, which I posted online, the old exams that are posted online, just the relevant parts of those, and then the parts of the lab manual that would be multiple choiceable. I would suggest looking through that stuff. Okay, with that said, then the rest of it's question and answer.
For plate Z on the pictures you have, what is the
Oh yeah, so plate Z is the one picture you don't need to know about.
Plate Y.
Oh, plate Y. Okay, plate Y on the pictures is the question. What's plate Y? I don't know. It's nothing appropriate. Nothing we've talked about. It's wrong. Or I can't think of an answer where plate Y would be the correct answer. Sorry, real quick. What other plates? There's nothing that we've shown you. WXU, fine. Those are crap also. Okay.
So plates or pictures K and P and Q. Because one of the questions is which one is a gram positive?
Right.
And it points to Q as being the correct answer, but that's a pink.
Yeah, that's.
Whereas it should be like, I would think it should be K.
So
or
which
P.
question is it?
It was asking about SEPI.
Number 5. So this is a common question. Number 5 on the old exam 1. So S. epidermis is gram positive. Just what is S. epidermis? It's a cocci. It's gram positive. It doesn't have spores. That's all true. So if you do the spore stain on S. epidermis, though, it'll be just pink because the only things you add to the spore stain is the malachite green and the safranin. So in this case, it will look like Q, even though it's gram positive because you didn't do the gram stain. It will only look gram positive if you do the gram stain correctly. So it's like kind of a trick question, but be careful which stain you're doing. It's in there because actually on many people's unknown projects, they'll say, oh my God, I swear it was purple last time, and now I did the spore stain, and it's totally gram negative. No, we did the spore stain, so it's different. All right, there's a question back here. I saw.
I was going to ask about M and N. It looks like they both have endospores, but one's been released and one's not. But like, if you were to ask which one was endospore positive, could either.
Right, so the question's about M and N on the image sheet, and which one is endospore positive. So yes, I would say this is probably like a younger culture in M, or a less stressed culture in M, and a more stressed culture in N. If these were both endospore stains, I would read them both as positive. So I would have to give you only one option probably. Or it could be more than one of the options listed are spore positive, maybe if I did it like that.
Can you go over question 7? It asks about like the volume of a cube, and it has like the units meters for each side,
right.
but then the answer choices are millimeters.
Yes. So question 7 is asking about like unit conversion between length and volume, and so the unit. What I always use for converting between different scientific units. Where is this? So the key fact that I remember to convert between scientific units is that 1 centimeter cubed, sometimes written as 1 cc, equals 1 milliliter in volume equals 1 gram if this milliliter was water under standard conditions. Okay, so. All right, so in biology and microbiology, we can assume that everything's watered. It's like the round cow in physics. You guys know the joke about that? Whatever. It's like calculate the terminal velocity of a cow, and it's like, we'll assume the cow is a sphere and in a vacuum. Things like this. Okay, so anyway, we'll assume everything's watered for the purposes of this class. In that case then, if we have 10 to the negative So there's different ways to do this using a calculator. I'm going to walk you through the way I would do it, but like I'm not a math teacher, and there's other ways to multiply this stuff and get the same answer. Okay, so what I would do is if we have 10 to the negative 1 meters, I would say that this is 10 centimeters, because it is. I would convert to centimeters though. And I'd say 10 centimeters times 10 centimeters times 10 centimeters equals 1,000 centimeters cubed equals 1,000 milliliters equals 1 liter. Okay, another way to do this, so my answers usually, I really won't ask you with answers like 1,000 milliliters or 1,002 milliliters or 1,004 milliliters. Usually be like a long ways off the different answers. So another way to do this would be to say, okay, 10 to the here's a meter. 10 to the negative 1 meter would be a tenth of a meter, so like this. If I had a cube this size, it'd be like the bottom half of a 2-liter bottle. So it's a liter. You should be able to sort of visualize these things also. To like ask yourself as you're pipetting, does this look like it could be 10 microliters? You should have like sort of a visual idea of what that would be. All right. So the key conversion then again is that 1 centimeter cubed equals 1 milliliter, and usually equals 1 gram. Is the assumption. All right. If we did another one of those, so let's do another one real quick. So let's say you do a microliter of water. How much should that weigh? So if we're working backwards, if we have 1 microliter of H2O, weight equals. So if we have, how much does a liter weigh? 1,000 grams per kilo. 1 liter equals 1 kilogram. So 1 milliliter equals 1 gram. 1 microliter equals 1 microgram. One way to figure that out by using I'm not sure, by using ratios, fractions I guess is how you can do that. Because this is divided by 1,000, that's divided by 1,000. Another way that we could do this would be to say that 1 microliter equals what part of a milliliter? 10 to the negative 3 milliliters. And so then we can multiply this times grams. So it would be 10 to the negative 3 grams. That would also be microgram. Sorry, I did this wrong. I wrote that. Yeah, and that's on video too. 1 microliter equals 1 milligram. That's because if you divide this by 1,000, microliters divided by 1,000 is milliliters divided by 1,000 is microliters, grams divided by 1,000 is milligrams. That's embarrassing. Okay. 10 to the negative third milliliters equals 10 to the negative third grams. That's how you can get a microliter equals a milligram. Okay, next question. Yes. So number 17 on the old exam 1 is asking about like a dilution factor, to calculate what's the dilution factor. So the easiest way to do this, or the like simple formula to use, is to make it a fraction that has the sample volume over the sample volume plus the diluent volume. So in this case, what it would be is, and we got to make sure we're in the same units. So in this case, we would have 5,000 microliters over 5,000 microliters plus 45,000 microliters. So that would be 5 over 50,000. Or 1, is 50K, or 1 over 1,000. Oh yeah, but that's not right. Yeah. How did I get this? 5,000. Oh, sorry. It'd be 5,000 over 50,000. I started getting simple here. 5,000 over 50,000, and that would be 1 over 10.
So when it says tenfold, should I think more like 10%?
A tenfold dilution is saying like this 1 to 10 dilution. So yeah. Yes, ma'am.
So number 24, the reversal of the crystal violet and the saffron stains.
Right.
The answer shows that all cells would appear purple, but if you added the colorizer, wouldn't it just be flipped?
So in this case, sorry, what number was that again? Number
24.
24. Okay, let me erase some stuff here. Number 24, it says, on the practice test, it says, what if you did first crystal violet? I'm sorry, first you did saffron. It says reversal of the crystal violet and saffron. And so in that case, it would be that first you do the saffron, and then you do the iodide, and then you do the ethanol, and then you do the crystal violet. So I'm not sure really what would happen between the saffron and the iodide, but it might be pink after the ethanol treatment, or it might be clear after the ethanol treatment. I could imagine it being either pink or clear. But then when you add the crystal violet, it's going to make everything purple again. Since there's nothing after that but just the water, everything's going to appear purple in this case. So it would be either clear or pink here, I don't know. But if you put purple on top of pink, it'll appear purple. Purple on top of clear will appear purple. So also keep in mind, I might describe it as pink, or I might describe it as saffron colored. I might describe it as green or malachite green colored, or purple or crystal violet colored would be synonymous to me. Right, pink and saffron colored are synonymous to me. Let me say one other way to do this question, especially since I managed to flow it. Instead of saying 5,000 microliters over 5,000 microliters plus 45,000 microliters, it might be easier to convert this all to milliliters. So how many milliliters is it? It's 5 milliliters over 5 plus 45 milliliters equals 5 over 50 equals 1. Yes?
So if that was, let's just say if it was 1 over 100, that would be 100-fold?
Yes.
Okay.
If it was 1 over 100, that would be 100-fold. So sometimes people say 100-fold, or 100x, or 10 to the negative 2, 1 over 100, 1 to 100. They all mean the same thing. So you might think if it's tenfold, it'd be 10 times as much, but then it would be a tenfold concentration instead of tenfold dilution. So we're always going to be doing dilutions in this class. Yes.
So for 23, it asks about like, the purpose of incubating an unopened plate, and I understand the purpose of it, but I don't understand why that's described as a negative.
Okay, so for number 23, so it's the unopened plates are controls, and there's 2 types of controls we run here, and negative and positive. Okay, fine. So which type of control is it? So in this case, I would call it a negative control because we expect nothing to happen, no change, and so that's where we get the negative control. And the positive control would be to make sure something can happen. So for a good positive control, we would check for growth. We'd put lots of bacteria there to make sure we could have growth, for example. So a positive control would be always looking for a change, and a negative control would be checking to make sure there's not a change. If we were doing this in for like a real life example, say you want to make sure that the bathroom scale is good for your travel bag to weigh in, make sure it's 50 pounds or less. So your negative control would be nothing on it. It should say 0. It should stay stable. A good positive control would be a bag of known weight, like a dog food bag is 50 pounds or something. And you want to make sure the scale does change appropriately. Yes.
The question for you, why is this?
For question 40, we didn't quite do this exercise fully, so I wouldn't ask, question 40. The basic reason why it's right is because in exercise 6.1, it says the most reliable counts are between 30 and 300. We didn't fully do exercise 6.1, so I wouldn't expect you to know that answer. I think you could rule out that A and E are probably not good examples. B, it turns out to be too close, still too many to count effectively, and D is less statistically reliable than C. Other questions?
What would the answer be for 22? It doesn't, on my thing, it doesn't have anything highlighted.
So when you get to the end of the exam, make sure that you have 40 answers selected on your answer form, because if you don't, you for sure get it wrong. So number 22, I overlooked it when I was making the answer key. The most likely answer would be A. That actually there's no universal media. There's nothing that can grow every bacteria. And when we measure, for example, how many bacteria grow in a petri dish, that's a really minimal amount that were present in the sample. Because there could have been others that grew but we didn't notice them. There could have been others that would have grown and had the conditions been right. There could have been others that would have grown eventually. Others that would have grown in cold temperature, hot temperature, anaerobic environment, high CO2 environment, etc., etc.
Could it be argued that it would be D because there would be likely that someone didn't cough on the plate?
Yeah. So you could make other reasons why. You could make other explanations for why. So this is a good relation. This relates to. 1 second. What's the most reliable result? Which is question 35. So there's lots of different ways to get a negative result. So for example, someone didn't cough on the plate, someone didn't open the lid, you accidentally put the plate in the refrigerator instead of the incubator, you didn't wait long enough, you didn't have stuff grow. But a positive result, that's much more reliable. Okay, so this is like my analogy for this is always bears in the woods. Okay, so how many of you guys think there's bears in the woods around Lawrence? Anyone? No one. Okay, so I bet you guys each $50 there are. All right, so now how are you going to claim your $50?
Maybe go out in the woods and see some fun bear.
But they're rare. They could be hibernating right now. I think they're probably nocturnal and camouflaged. And they're actually today they're where you were yesterday, etc. But then one day when there's a bear, you guys each owe me $50. And if you want to talk about bad incentives in science, at some point there's going to be enough money out there for me to buy a bear. Drag him in the woods and win my $50. So anyway, the point is that a positive shows something. A positive is a change, and it's much more firm that there was something than a negative. The fact that there was nothing observed, no change observed, is a lot weaker evidence. All right. And so the only way you can win the bet would be to say, I bet you can't find one in 48 hours. Something like that. And that's what we call a negative result. We say no growth or no color change in 48 hours is a negative. Lack of presence of spores is a negative. If you do 2 slides and can't find spores anywhere, they call it spore negative. We didn't really test that. It's much more certain it's spore positive if we saw spore. Yes?
Can you see one of the other dilution problems from the quiz line?
Yes.
Number 10.
Yeah, so if we look at, for example, number 9 and 10 on the quiz, there's a trick that we can use. So I'm trying to think of how to explain why the trick works, and I'm not sure. But let me show you how the trick works. All right, so let's say we got 1,000 cells. All right, and then we kill 90% of them. So kill 90%. How many survivors are there? 100. Okay, and we kill 90% of them. How many survivors are there? 10. And we kill 90% of them. Well, 1, right? Okay, so it turns out then, if we start with 1,000, what's 1,000, and we kill 99%, we get 10. And if we kill 99.9%, we get 1. And it turns out that if you kill 90% twice, it's the same as killing 99% once. This is 99.9%. Okay, so it turns out that each time you kill 90%, you move the decimal point over 1. So what the trick is on the quiz question like number 9 is if you have 2 times 10 to the 8th bacteria and you kill 99.99%, then either what you can do is you can take, you can make this into like an integer, like take 100% minus this, and it's going to be 0.0001. You can multiply that times this number. Or the trick is you can just count the number of 9s, 1, 2, 3, 4. You subtract that number of exponents. And so it's going to be 2 times 10 to the 4th. And it just works like that. All right, so I would just say 2 times 10 to the 4th is the answer there.
You mean that's how many are still alive?
That's how many survived, yes. So here, for example, you start with 1 times 10 to the 3rd. We start with 10 to the 3rd, let's say. And then you killed 99%, so you went down 2, 1, 2. You're left with 10. Okay, so what about number 10? Number 10 is slightly more complicated. So we had 10 to the 6th CFU per mil. We killed 99.9%. This is number 9. Number 10, 10 to the 6th, kill 99.9%. So that leaves us 10 to the 3rd cells, right? That's 10 to the 3rd. But what is it? What's our units? We need to be careful always to take our units with us when we do math. So it's 10 to the 3rd CFU per milliliter. And it says in the question that we only plated 1/10 of a milliliter onto the plate. And so that would mean equals 10 to the 2nd CFU on the plate. Or 100 colonies on the plate. So 10 to the 3rd CFU per mil is then the population in the test tube. Okay, so we got first start with 10 to the 6th in the test tube. 10 to the 6th CFU per mil. And then we killed it, so I'm going to shine a light on it. This is the light shining here. Okay, and now we end up with the same test tube. Then it becomes 10 to the 3rd CFU per mil. And what we did then was, according to the question, is we put 100 microliters, which is 1/10 of a milliliter, out of this tube onto this plate. And so then we would expect 10 to the 2nd CFU to grow. And so what we're doing is moving down in order of magnitude because we're only using 1/10 of the sample. So imagine you have $1,000 and I take 10% of it. So even though I didn't dilute your money any, but I only took 10% of it, so I only get 100. Next question. Yes.
How did you know to go from 10 to the 6 to 10 to the 3rd? That's.
Because I counted how many nines there
Oh,
were.
okay, got it.
99.9%, there's 3 nines, and so I can go from 10 to 6 to 10 to the 3rd. The other way to do this would be to multiply and calculate. You take like 10 to the 6th times 0.001, right? And you get the same thing. And this would be moving over 1, 2, 3 decimal points. So you get the same net effect.
Let's go over question 10.
All right, number 10 on the quiz.
Yeah.
I just did that.
Sorry, the practice.
Well, the practice exam. Okay, yeah, so number 10 on the practice exam is just asking asking you to backtrack to say what your original CFU per mil was. This is sort of like what we did on practical 1. So let me erase a little bit. Okay, so it says that we did 10 to the something dilution factor. So that means we're just going to be moving the decimal point over. So let's see. Now, number 10 on the practice says that we ended up counting 24 colonies, 24 CFU on the plate. Okay, so how many CFU were in the sample? It says that we only inoculated 1/10 of a mil onto the plate. So then that would be equals 240 CFU per milliliter in the sample. And the sample, it says, was a 10 to the negative 4th dilution. So to correct for a 10 to the negative 4th dilution, then we have to multiply both sides by 10 to the 4th and move the decimal point over 4 spots. So I would say 240, 1, 2, 3, 4, and it would be 2.4 times 10 to the 6th, or 200, or 2,400,000.
I'm sorry, how did you get from 24 to 240 again?
24 to 240 was going from the the number of colonies observed on the plate to going to the number of colonies that must have been in the sample. Because it says that 100 microliters were observed, or 100 microliters were applied to the plate to get that number of colonies. So again, this is like saying this is how much was in your pockets, and when we took 10%, asked you to show 10% of it, we came up with 24. So we're just taking a sample out of the mill. And generally, it will always be 100 microliters that we plate in class, and it will always be 100 microliters that's used on these platings in the math examples. So that's 1/10 of a mil.
So if it were to say like we inoculated to like 1,000 microliters, it would then be 200 for that, or 2,400.
If it said that you inoculated 1,000 microliters, then the actual number would be 24 CFU on the plate. If it said that you did 10 microliters inoculated, then it would be 2,400.
Oh, okay. Okay.
It's just saying how much of the sample how much of a milliliter was taken. Because our standard thing that we talk in, or our standard units that we're going to talk about bacterial populations in is CFU per mil. And then we're usually transferring microliter amounts.
If I'm getting this right, 100 microliters equals 1/10 of a milliliter.
Yes, 100 microliters is 0.1 milliliter. And so it acts as if it's one more dilution factor, because we're just taking a small sample of them.
What's the difference between spore positive and spore negative?
So spore positive, we would say that we for sure have spores present. Spore negative, we would just say no spores were observed. So this is like the bear thing. You can come back today and say I didn't find any bears last night, but you can't really be sure there weren't any. Other questions? If you have questions during the exam, so if you happen to have any questions about the images, like say, is this pink or purple, is this a rod or a oxide, you can ask that in advance of the test and we can be really clear answering it. If you ask during the test, we can't answer that at all. So ask in advance, things like that. If you have questions about the wording or the phrasing on the questions or answers during the test, you can ask your TAs. The best way to get help on this, though, is to offer like a synonymous phrasing, offer a rephrasing. Does this mean, would this be the same as this? And try to give them an alternative phrasing. Because if you say, what does this word mean? It's really hard for us to say. If you say, does saffron mean the same as pink colored? Then we can say, oh, yeah. Just say, what does saffron mean? Which can't do. All right? Great. Thanks for coming.
Thank you.